Hello, everyone. Welcome to the first ARC seminar of the year 2025. We have two very exciting talks today. One on deep quantitative and affordable paired TCR repertoire sequencing, and the other one on a pipeline for inference of phylogeny and selection pressure in BCR sequences. The format is as follows: that the first talk on here now today is sequencing, TCR sequencing will be 45 minutes plus 15-minute questions. And then afterwards, we have a shorter talk of 20 minutes plus 10-minute questions so that we kind of finish in 1.5 hours roughly. And all of the videos and the recording will be found on the YouTube channel of the AIR community. So I will introduce the first speaker, Mikhail Pogorelyy. He is the Director of Computational Science at St. Jude's Children's Research Hospital and specializes in adaptive immunity and T-cell receptor repertoire analysis. His research focuses on creating experimental and computational approaches for deep paired TCR and BCR repertoire sequencing to unravel immune system dynamics in cancer, immunotherapy, gene therapy, and infectious diseases. And for example, during he has many publications, but for example, during his postdoctoral training in the Paul Thomas lab at St. Jude's, he developed a reverse epitope discovery approach to identify TCRs, recognizing novel epitopes just from repertoire sequencing data. And of course, his PhD was performed in Moscow in collaboration also, for example, with the Walczak and Morat lab in Paris. And today he's going to talk about a very exciting preprint, TIRTL-seq, and I think we're all eagerly waiting for your talk. Thank you very much for being here.
Thank you so much, Victor. Yeah, I want to thank the organizers for letting me talk today. I'm directing this initiative at St. Jude we call TIRTL. I frequently forget how exactly it spells out, but it's all about developing new repertoire technologies and applying it mostly to pediatric cancer and infectious disease problems we deal with here. So we will talk about TCRs today. I will do very brief introduction to TCR. T cells are key players in adaptive immune system. They recognize pathogens using their T cell receptor. There's a lot of potential pathogens. So you need a lot of T cell receptors. And because of this, T cell receptors are not encoded in genome in their final form, but are formed in semi-random VDJ recombination mechanism. So in genome, you have these cassettes of segments, you join them, you delete and introduce random nucleotide at the junction. Receptor is heterodimer, there is alpha chain and beta chain, and both recognize the antigen. The antigen is recognized bound to MHC complex. So on the left, you see the structure. We're looking at it from the side. TCR is on top, and this is peptide bound by MHC. And very brief, like primer, how it works in practice. All our cells constantly chop peptides they make and present them on MHC complex. If cell is infected by a virus, right, viral peptides are also synthesized, chopped, and presented. All T cells after this repertoire generation process, they go through selection, which depletes T cells recognizing self. So if recognition happens here, this means that this is not self. This is probably a viral peptide. And then two things happen. This T cell, which recognizes the antigen, it proliferates, and all the progeny has exactly same TCR and forms a clone. And it's also differentiated, gets a function. And in this particular like viral infection case, you might imagine the function is just cytotoxic. So it kills the next cells which are infected with virus in the organism. So this is just very simplified. And of course, one way this system works. And this is a textbook picture. We will need some additional facts, right? So one fact is from structural studies, we know that both TCR alpha and TCR beta chains make contacts with peptide MHC, right? So this is from one of the Chris Garcia's reviews, right? So you look on the PMHC complex from the above, and you see this is a footprint of V beta, this is a footprint of V alpha. Both of them make contacts with this peptide here. And this is the same interface as here, but from the side. This is critical because TCR beta, TCR alpha or TCR beta by themselves do not determine specificity. If you know just TCR beta, you're very limited in what you can do with this TCR, right? So you could not express it and test it against different antigens, right? You might compare it to some existing TCR and argue, okay, maybe if the beta matches, specificity also matches, but it's very hard to test it, right? To test it, you really need a second chain first. So having two chains is important. And another important fact is that this is quantitative, but relevant immune responses occur very deep within T cell receptor repertoire in most cases, right? So very early on, people figured out that T cell receptor repertoire clone size distribution is power law. This means that you have very small amount of big clones and a lot of very tiny clones, right? So usually your biggest clone is 1 to 10% of all your T cells. And then you have 10 clones, which are 0.1%, and you have 100 clones, which are 0.01%, and so on. As you go deeper and deeper, right, amount of clonotypes you see at this clone size level increase. And in almost any T cell sample, majority of clones are just found in a single cell. And this applies to naive, memory, PBMC, any T cell sample you could think of. And then there is an actual immune response, right? So what we know is even at the peak of the response, only a small fraction of your T cells recognize any given antigen. So this figure is from the classic paper from Rafi Ahmed group, Rafi Ahmed lab. It is yellow fever vaccination. Yellow fever is a model of acute viral infection. It's a very immunogenic vaccine. It's a live virus, in fact. And here they stain with HLA-A2 tetramer, which stains all the T cells recognizing certain immunodominant epitope. In the bottom, this is controls. These are HLA-A2 negative people who could not develop response to this epitope. And on top are HLA-A2 positive vaccinees. And you see that, and this is around the peak of infection, and you see that just, maybe up to 0.5% of the T cells recognize this epitope. This is typical, right? In some cases, you have more, right? But what we need to understand that this is not a single clone. So this is 0.5% of T cells, but this is probably hundreds and thousands of clones combined. And if you look on the single clone level, and we did it by next generation sequencing in yellow fever context and in COVID context as well in several studies, you see the typical size of a clone recognizing viral infection at the peak is like 1 out of 1,000 cells, 1 out of 10,000 cells, right? So it is relatively, so this is relatively rare events, right? But the combination of them, of course, form something like few percents of your repertoire. So what toolbox we have to study T cell receptor repertoire? So there are more tools, right? I just list the most frequently used ones. You have, of course, bulk TCR repertoire sequencing. It is deep, but it's a single chain only because when you do it, you lyse the cells. Alpha and beta are encoded separately, different chromosomes, different transcripts. And when you lyse them, they all mix. So you don't know which alpha was paired to which beta. It is reasonably cheap. It could be done from RNA or DNA, and this is just an estimate if you do it yourself. I think Adaptive charges you way more, which is the most frequently used company for repertoire sequencing. So about $200 per sample, and sample could be, I don't know, 10 million cells. You can sequence 10 million cells with this technology. And another alternative is single cell TCR sequencing, which is a subset of single cell RNA sequencing, where you enrich for TCR alpha beta cDNA in your library. And this is figures for 10x genomics, about $2,000 per sample, and sample is like 10 to 20,000 cells, right? So you have paired TCRs. You also have phenotype, which is cool, but very limited amount. And given what I told you, but in some cases, viral infected, so viral specific clone is just one cell out of 10,000, right? So this is about the same scale. So ideal technology would be something low cost as bulk, high throughput, because we really need to analyze millions of cells to understand what size the clones are and to track them maybe in time. And it should also be paired, because if you know just one chain, you could do almost nothing with it experimentally. You're very limited in the next steps and figuring out what this TCR really recognizes. So I will give you like our solution, right? But before that, let's think about what makes single cell RNA-seq technology single cell, right? So single cell RNA-seq is, of course, a revolution in biology, right? It's used everywhere now. But how does it really work, right? So, and there are just two principal solutions. First idea is, but if you could do bulk RNA-seq and you could put just one cell in your tube, right? You have your single cell RNA-seq solution, right? But then you need a device which could manipulate a single cell, right? So I call it like mouth pipetting, but the idea is that you could use sorter, which put like one cell per well. There are like single cell dispenser machines, but you move single cells physically. Throughput is low, and it depends on an instrument, of course, right? So rarely you could do more than a few hundred cells this way. And another idea, which is behind all the high throughput single-cell RNA-seq technologies we have today, is the idea of limiting dilution. You don't move individual cells. What you do instead, you have a cell suspension, and you count them, so you know the density of cells in this suspension. And then somehow you create a lot of containers, in many cases millions, right? And you put your cell suspensions, you distribute it across these containers. And on average, each container either have one cell or it's empty, right? Because your suspension is very diluted. So this is this limiting dilution principle. And different single cell technologies, they differ in a way how they create this crazy amount of containers to put your cell suspension in. So you could think of a simple solution like, okay, let's just get multi-well plate, like we have 384 well plates, I don't know, 1536 well plates, and just distribute our suspension there. But you could understand that throughput gonna be very low, right? If half your wells are empty, right, that's not much. You could miniaturize a well and make it like a teeny tiny well on a chip. And this is how BD Rhapsody works. You could think of a container as not a well, but as an emulsion droplet. And then, of course, you could have a lot of those. You barcode them with special beads. And this is how 10x genomics works and other technologies like this. But I think the most conceptually interesting idea is that container is a journey, not a destination. And this is what happens in split-seq, commercialized by Pars Bio. And the idea of this technology is they have their cell suspension, they split it, they dispense it into 96 well plate. When they introduce a barcode, they pull the cell without destroying the cells. They pull the cells back, they split them again, they introduce another barcode on top of first barcode. They do this several times. And after that, each cell is labeled with the string of barcodes, right? And you could understand that there's a lot of this potential strings, right? There are a lot of journeys each cell could make over all this containers. And this is basically your container, although you have just 96 wells, right? So if we talk about T cells, I told you that T cells are clonal. We could abstract this idea of container even a bit further. And we could say that container is a combination of wells in a plate, right? Not a single well as a container, but a combination. And then it turns out, and I think the math here is like cool and a bit counterintuitive, but there are a crazy amount of ways to pick, let's say, 4 wells out of 96, right? So here I just show 3 different options, right? So this is like this blue container, red, and green, right? But there are like 3 million of those. And this is just like n choose k, right? So there are a lot of potential containers. And then imagine you have your T cell sample, maybe 10 million cells, right? And you distribute the sample equally. You split it between all these wells in 96 well plate. And then, as I told you, most clones are small. So there are quite a bit of clones which are which have exactly 3 cells or 4 cells. And it's very likely that if clone has 4 cells, it will end up in 4 different wells in this plate. I mean, of course, you could be unlucky and 2 cells end up from 4 cells total, 2 cells end up in 1 well, but in general, it will distribute across 4 different wells. And one idea is that you just run bulk. You run bulk TCR alpha and TCR beta repertoire sequencing from each well. And then if you look and find certain TCR alpha is found in particular 4 wells, what you do next, you look for TCR beta, which are found in the very same wells. Right? So this is the idea of combinatorial TCR repertoire pairing. Right? So you get your cell sample, you split it into many partitions, right? 96 minimum. And then you determine patterns for alpha chain, like well-occurrent patterns, where I see a certain alpha. And you find matching beta chain pattern. And this idea was suggested by Brian Howey in Science Translational Medicine paper in 2015. It was originally developed by Adaptive Biotech. They got very good results with it, but the whole technology wasn't practical because it was very expensive. So this is today's cost. Back then it costed more, but I estimated about more than $2,000 to process one 96 well plate. And also it took like a few days of library prep with all the cleanups. But they get like more than 100,000 paired TCRs with this technology. And then if we think about this mechanism, this algorithm, right? So there's also obvious limitation. If something is found in very few wells, like in 1 well or in 2 wells, we could not pair it, right? The pattern is not, clear enough. There are just 96 ways to pick 1 well, and then there are millions of clones which are found at 1 cell level, right? So 1 well is going to be a mess. We need at least 4 wells at 96 well plate design. And then again, if the clone is really big and it ends up in all wells, and there are a bunch of clones like this, again, you couldn't pair it, right? So if all wells are occupied, there is also no pattern to match, right? So in this sense, it is symmetric. And we were inspired by this idea, and we developed a TIRTL-seq protocol. And the idea of this protocol is that we we increased number of wells, and we aggressively miniaturized all reactions, and we also excluded majority of steps from original protocol. So all the cleanups, all plate-to-plate transfers, all of this was sacrificed, and the protocol still works. From molecular biology perspective, you have this 384 well plate. In each plate, there is a lysis and RT solution, and you load cells directly. So there is no extraction step. You get your cells and you load them directly into these wells. You use non-contact liquid dispenser for this because 384 well plate, it's tough. And with machines, they basically produce small droplets with your reagent, put them into each well. And then we run oligo-DT and random hexamer RT reaction, cDNA synthesis. And then on top of that, we have PCR step with V multiplex primer and primer specific for C segments of TCR alpha and TCR beta. And this is just overlaid on top of RT reaction, right? So you just pipette things into the same tube. And then you run another PCR when you barcode each well, right? And you pull it all. And in early versions, we use salad spinner to do this, to pull all the libraries together. There's a single beat cleanup, and you do sequencing, and you get ready to sequence library afterwards. So the whole process takes maybe 5-6 hours, and hands-on time is really small here. And the cost per one 384 well plate for reagents is less than $200. You need quite a bit of equipment to run this, right? So you need non-contact liquid handlers, and you also need something to do a plate-to-plate copy to transfer PCR1 into PCR2. If you don't have it, we also developed manual 96 version of protocol, which has same cost. It is half more efficient in terms of pairing because, I mean, you lose some resolution when you drop from 384 to 96 wells, but you could do it if you just have a multi-channel PCR machine. And the idea is the same, right? So you don't extract RNA or DNA, you have your cells, you pipette them into plate containing RT solution, you run RT, you pipette PCR1 master mix on top, transfer it to a PCR2 plate, and you're done. And you could compare it to 10x Genomics Chromium platform, which is $2,000 for 20 1,000 cells and like 3-day library prep, right? So it's like 10 times cheaper. So we developed this protocol for combinatorial pairing to work. Two things should be true. First, the protocol should be sensitive down to single cell level. You should be able to notice presence or absence of a single cell from a clone. If you lose cells, right, in this process, your patterns are getting blurry and it's hard to match TCR alpha to TCR beta. So first thing we checked, we did this protocol, but with sorting just one cell per well with a sorter, right? And here, if it's green, it means that we retrieve both alpha and beta. If it's empty, there is no cell, right? And sorters are not perfect. Sometimes we miss, right? And cell gets stuck somewhere, or there is no cell in the droplet. So we know this from other technologies as well. This is control column, it's empty, so no contamination issues, this is good. And then there are a few wells when we lost either alpha or beta, but very, very few. So in general, technology is very sensitive down to single cell level. And another idea which makes combinatorial pairing really efficient is that you need to put a lot of cells in each well, right? You need to put a lot of cells in each well because more cells you process in this technology, more pairs you will get. And we tested this as well. So we loaded from 12,000 PBMCs to 100,000 PBMCs per well. And here we just plot number of unique clones we get. There are some artificial diversity. You see we loaded 12,000 cells, but got like 20,000 clones. And this is, of course, due to sequencing errors. But in general, you see the trend. You add more cells, you get more clones out. But for alpha, it saturates. Around like 50, you don't have such a good increase. And we also see that pairing efficiency drops. So basically 25,000 cells per well is optimal, but this is a lot. So 25,000 cells per well, if you have 384 well plate, this ends up being 10 million cells per 384 well plate. And what we also found is that if you upscale RT and PCR reaction volumes, then you could also increase number of cells per well proportionally. For manual protocol, we upscaled it 4 times and we put about 100,000 cells per well, and it works efficiently, right? So if you have more cells, you could do this, but I will show you a better way to deal with it, right? So we have a protocol. How do we actually execute the pairing? How do we analyze data? Original Howie paper didn't share any code. They give kind of an outline of computation, but it was not detailed enough to reproduce. In 2019, Michael Birnbaum group released this MetHype algorithm, which was reanalyzing Howie data. It's a Bayesian framework to pair TCR chains from these combinatorial experiments. Idea is that it just gets 3 integers to get a pairing decision for a given alpha and beta, right? So result of combinatorial experiment like this is like millions of alpha chains and millions of beta chains. And then you need to compare each alpha to each beta and try to answer if they're paired or not, right? And I told you, oh, you simply look for the beta which is found in same wells. This is true, but in reality, you have some loss, right? So sometimes you lose chain and don't detect it. So 3 integers are number of wells where you detect both alphas and betas, both this given alpha and this given beta. Number of wells where you find just alpha, right? And if clones paired, this means that these are wells where alpha is lost, right? And then, of course, there is number of wells where you detect just beta, so you lost alpha, right? And ideal situation, of course, if you have big overlap, so you always found chains together and you have no loss, so chains are never found by themselves, right? But for a given design, you need the model to answer that. Original software was very slow. It took 24 hours or 96 CPU cluster to analyze one experiment. So we spent a lot of time to optimize it algorithmically. But it turned out the biggest optimization you can do is not trying to make a better algorithm, but express everything as tensor operations and compute on GPU. And basically this 24 hours or 96 cores end up being 93 seconds on a single GPU for our implementation. And this really helps because our ambition was to increase number of wells, right? So at base we increased it from 96 to 384, but you could push it even further. So it was very important for this computations to be efficient. I would like to touch like just a little bit on the critical algorithmic improvement here. So critical algorithmic improvement we did was using a lookup table basically, right? Because entire algorithm gives you pairing decision based on these 3 numbers. right? And how many wells you find them together, and how many wells you find alpha and beta separately. So idea is you could just pre-compute it all, right? And this is how it looks like. So this function takes 3 integers and output you a pairing decision. And if it's red, we reject the pair if there's a big loss, right? If it's blue, we accept the pair. And you see this is like, this exists in this simplex, and there is this zone, and there's a 2D border of this shape, right? And within this shape, we accept everything. And outside this shape, we reject everything. And because there's a 2D border, you could really translate this 3D decision simplex pre-computed into 2D table. And basically you ask this question: for each overlap value, if I have certain loss of beta, what is the maximum loss of alpha I could tolerate, right? And if you could answer this question, and the answer is like this 2D table showing this maximal loss, right? Then you could speed up the computations dramatically, right? Because below this loss, you will accept every pair, and above this loss, you will reject every pair, right? So yeah, this is very small and technical detail. So let's get to the actual results, right? We got PBMCs from a healthy donor. We isolated CD3 positive live T cells and did conventional 10x genomics single cell TCR-seq on it. We loaded 20,000 cells. After it, we got 11,000 cells with paired complete alpha beta TCR, which corresponded to 9,000 clones. And this is this line. And then this costed $2,000, and we did a TIRTL-seq 384 well plate, which cost $200, and we loaded 25,000 PBMCs per well, so 10 million total, and we got about 170,000 pairs from it. And then, of course, you could downsample. You could imagine if I only analyze 96 wells, and you still are better off with the TIRTL-seq protocol than you are with 10x. And because it's so cheap, idea is that we could do we could add additional plates, right? So we did 1 384 plate. If we have more PBMCs from the sample, let's do more, right? And as you add additional plates, I will not go too much into detail, but there are 3 different ways to integrate the data, right? So you could analyze all the plates separately, for example, and just merge the results. Or you could kind of stack them on top of each other, right? So A1 well from the new plate is combined with A1 well of a previous plate, and this kind of mimics what happens if you just add more and more cells into 384 well plate, right? Or you could imagine I have like 768 plate now, right? So you just increase and analyze bigger plate altogether. And of course, as you could imagine, this increases the resolution of the pattern, and this is the best way of scaling when you analyze bigger and bigger plates. So while scale gonna change here. So this is what you will get at 2 plates, 3, 4, 5, 6. At 6 plates, which is like more than 2,000 wells, we hit 1 million unique paired clonotypes, and the overall cost is still below what you get with 10x genomic sequencing. And this 1 million unique clones are corresponding to at least 30 million cells, right? Because each clone, at minimum, right, is found in 3 wells to be paired, and a bunch of clones are found, of course, in more wells because we're more expanded. And here is some estimate how much it will cost if you do it with conventional available single cell TCR sequencing approaches. But then the question is, we identified all these pairs with our algorithm, but is it correct, right? And this is why we did 10x in the first place. We wanted to validate if we pair clones correctly, right? Because it might be that you get 1 million clonotypes from the algorithm, but I don't know, half of them are just wrong pairs, some kind of artifact. So what we do here, this is what we see in 10x genomics experiment, right? So you see largest clone is more than 100 cells. This is second largest, third largest, and so on. So this represents clonotypes from 10x genomics experiments. And I color them with red if we were able to pair it in this TIRTL-seq experiment. And there is good news. Good news is that out of clones we were able to pair in TIRTL-seq, 92% has exactly same pair. So precision is good. Bad news is that all TCRs we were able to pair are less than 5 cells big in 10x genomics experiments. So we completely miss all these big clonotypes, right? So 10x pairs big clonotypes. We could not pair big clonotypes. And the reason is simple. We loaded about 20,000 cells in 10x and 20,000 cells in each well. So basically, if clone is 5 cells in 10x, it is likely to be found and sampled in almost all wells in our TIRTL-seq experiment. So there is no pattern to match, right? So this is the situation when you have clone sampled in all wells, right? And you could not match alpha and beta patterns for large clones efficiently. All of them kind of look the same in this assay. But when we got an idea, right? So if clone is sampled in all wells, there is still low count noise, right? So if clone is in all wells, some wells got 5 cells, some wells by chance maybe got 1 cell. And the idea was that we could try to extract this signal, right? So if you have 5 cells, your relative frequency, if you just look on, fraction of reads occupied by this clone in this given well, it should be 5 times higher than in a well which got just 1 cell. So basically what we do here, this is for biggest alpha in the repertoire by average, and this is for the biggest beta in the repertoire. And this is just correlation of relative frequencies across wells, right? So we have like read percentage for this beta in each well, read percentage for this alpha in each well, and we try to plot correlation. We see no correlation if we compare alpha 1 to beta 1. Here we compare alpha 1 to beta 2, same, no correlation. Alpha 1 to beta 3, no correlation. Alpha 1 to beta 4, boom, we have very significant correlation, 10 to the minus 38, R 0.6. So this is just Pearson correlation. And you could continue doing this. And of course, you overlay all necessary multiple testing corrections. And this is minus log 10 p-value. You see that for this pair, with alpha 1 to beta 4, it's gigantic. For rest, it's below the multiple testing correction threshold, and you have 10x, so you could validate this is indeed correct answer, right? And we call this algorithm T-shell, and it fixes crucial flaw in this combinatorial pairing algorithm, which is inability to pair biggest clones. And in many cases, you care about biggest clones because these are expanding cells, which probably recognize current antigen. And then if we combine both T-shell and MetHype algorithms, we see that we cover now the big clones as well. And this is just 1 plate. We did 6 plates. You could extend it. Again, you see that vast majority of clones matches has exactly same pair between 10x and TIRTL-seq, and you also extend coverage a bit. Yeah, and this is just representation of the same, right? So this is comparison between 10x and TIRTL-seq and amount of pairs where you have exactly same matching alpha beta pair between 10x and TIRTL-seq. You see it's around like 90%. But if from 10x you exclude singletons, precision grows, precision goes up, right? And basically this is a hint that maybe if you have just 1 cell in 10x, frequently it might be incorrect pair. It might be a sequencing error, it might be an artifact. So basically idea is that maybe TIRTL-seq is even more precise, and these mismatches we have to 10x are mostly concentrated in a single cells found in 10x, and these single cells in 10x are not correct pairs. So it's bad on it might be bad on 10x side. And we could do some other sanity checks. For example, we could look how many partners different alphas and betas have, right? Because of course, in general, we want like, 1 partner. But from biology, we know this is not always true because cells have 2 chromosomes and 2 alphas are recombined at the same time. So in like 25–30% of cases, beta clonotypes have 2 alpha partners. And this is somewhat what you expect from biology as well. And there are way less cases when alpha chain have 2 beta partners, right? So most alpha chains are paired to a single beta. And again, this is kind of what you expect from what we know about TCR recombination. So this is nice, right, to have pairing, but the question is, how quantitative is it? And could you really use it to track TCR repertoire in time, right? Because we sample a lot of cells to identify clone sizes precisely, right? And if we do it at several time points after acute viral infection, we will be able to find clones which go up and then go down after infection is resolved, right? And this is what we know about T-cell response in general, right? So there's clonal expansion, when there is a peak virus infection clearance, and when there is a contraction pattern, when these T-cell clones are not needed in this amount and they die off and go down. In this case, we use the donor from cohort called St. Jude Trace. This is an employee cohort started here at St. Jude. Early in pandemic, a lot of employees were sampled. When everyone was tested, PCR tested for COVID biweekly, if person was positive, they were given more samples on acute phase of infection and then one at convalescent. On each sample, we performed 5' raise bulk TCR sequencing of alpha and beta. So this is conventional bulk TCR-seq approach. We did TIRTL-seq, and in this case, we split into CD4 and CD8, and we did half a plate, 192 wells CD4, 192 wells CD8. And we also did a 10x reaction, one 10x reaction per time point to compare all these methods. And basically, TIRTL-seq, it is like hundreds of replicates of bulk repertoire. And what you can do to figure out the clone frequency, you could just average clone frequency over all these wells, over all the replicates where the clone is sampled or not sampled. And after this average, you have this dots. This is in log scale. This is frequency of clone, average frequency of clone at baseline plate. This is average frequency of clone at acute plate, right? And what is not shown here, this averaging also gives you an error bar, right? Because if you average over, let's say, 100 values, central limit theorem starts working, and you could tell what is the standard error of the mean in this case, right? And basically, you could imagine there's a tiny error bar attached, I mean, in some cases, tiny error bar attached to all these clones. And if error bars do not overlap, you could say that clone between time points, that clone is expanding or contracting, right? So basically, doing hundreds of replicates makes statistical testing for clonal expansion and contraction between time points also very straightforward, right? And here I don't plot error bars, but I do colors, right? So this means that clones are significantly expanded. This means that clones are significantly contracted. And you see that there's quite a bit of expansion. So there are clones which are very big on acute, almost absent at baseline. And then if you compare acute to convalescent, you see that mostly it's dominated by clonal contraction. And this is the case for both CD4 and CD8. And this is somewhat what you expect from this picture. But if you do it with 10x, right, you don't get anything like this because you just sample 20,000 cells. And as I told you, right, so I mean, you could see some of these gigantic expansions, of course, but for these contractions, you miss this signal almost entirely because this is happening in this 10 to the minus 4 range, right? And for 20,000 cells sampled in 10x, this means 1 or 2 cells, right? And with bulk, it's more comparable, right? So bulk is just PBMCs, but you see this looks very similar to this. And if you look on the expanded clones identified in bulk and identified by TIRTL-seq, you see very good overlap, right? So basically, conventional bulk 5' race and TIRTL-seq applied to longitudinal data find same expanding, contracting clones. And then frequently people use combination of bulk and 10x to get both expansions, contractions, and pairings. And we can compare TIRTL-seq in this scenario, right? So you get your expanded clones from bulk sequencing, and then you look up in 10x or in TIRTL-seq, done on all 3 time points, to find the pairs, right? And again, we see that TIRTL-seq beats 10x because number of pairs is bigger. And this is another figure which illustrates another interesting point, right? So I showed you before this test experiments with PBMCs when we got a lot of pairs, right? So this longitudinal sampling, it's more like real life scenario because we do cryopreserved samples, we isolate CD4, CD8, we don't do full plate, we do 192 wells per sample. There are like issues with viability and cell numbers. We got like about 2 million cells instead of 10 million, right? So number of pairs we got for CD8 is actually lower. It's about like 10,000, 15,000 on each time point, right, for TIRTL-seq. And we got way more for CD4. So combined, CD4 and CD8 beats 10x, of course. But one might say, okay, you have like 15,000 pairs for CD8 TIRTL-seq. I mean, if I sort CD8 cells and do 10x, I will get the same. And this is not correct in the sense that you may get the same amount of pairs in 10x, but the clonal reproducibility will be lower, right? So because TIRTL-seq pairs clones which are found at least 3 cells, right, it is biased to pairing more expanded clones, right? And you process more cells in general. 10x could pair a clone which is found in a single cell. And actually, most clones paired by 10x are found just in a single cell, and you will never see the same clone again, right? So what is shown here was like red sector is unique clones, which you detect only in one time point and don't see it again. So we did an expensive experiment. We did 10x on each time point, and like 92% of clones were never seen again, right? And for TIRTL-seq, I mean, 50% of clones you find in our time points. And of course, if you want to find expansions and contractions, 10x will not work at all, but bulk repertoire sequencing and TIRTL-seq give comparable patterns in terms of expansion contraction numbers. And then because we have both chains, TCR alpha beta, we could make some next steps, right? We could, of course, compare this paired data to VDJDB and use TCR dists to find some matches. And we identified indeed that there's a bunch of clones which are similar to known COVID epitopes, and they expand here from baseline to acute, and then they contract from acute to convalescent, as you can expect. But then we were up to surprise because we found that there are quite a bit of matches to EBV TCRs, and these are the biggest expansions. So this biggest expanding clone is not COVID specific, it's EBV specific. And what's interesting, if you compare acute to convalescent, this clone does not contract, it's here to stay, right? So COVID clones go up and down, EBV clones go up and don't go down. And this is just total, this is just sum of all matched clones, right? So you see this difference in dynamics. And because we have paired TCR sequencing in this case, right, we could make a next step and actually validate these predictions. We cloned 8 TCRs, we picked 2 TCRs for each epitope, and we tried to pick like largest responding TCR. And then we co-cultured them with cell line which expressed just a single HLA, and we pulsed it with peptide we think the cell line should recognize. And our Jurkat cell lines are NFAT GFP cell lines. So if TCR is activated, we express GFP, right? And this is how it looks with media. This is how it looks if you add cognate peptide, right? And this is quantification for all these 8 cell lines. You see nothing in control and a lot of activation when target peptide is added. And one of the cell lines, 2 of the cell lines actually recognize cross-reactive epitope, which is cross-reactive between common cold coronaviruses and SARS. And then also we did serology to confirm this idea that probably this donor was co-infected actually with EBV and SARS, right? And we see that indeed at baseline, it's seronegative for EBV. At acute and convalescent, you have a lot of anti-EBV IgG, and same for SARS, but for COVID, we get PCR test. So this was somewhat expected. And natural question, of course, is could we do it for BCRs, right? So we do it for TCRs, could we do it for BCRs? There are unique challenges to this. I mean, it's work in progress. We're doing it for BCRs and it's working. I hope to maybe present on it somewhere later this year, but B cells are rare, right? It's just 5% of PBMCs, so you need bigger samples to do something. B cell clones diverge due to somatic hypermutation, right? So you probably need to change analysis for clonal trees. It's also nice to have full-length BCR sequence to sample all these hypermutations, and our current sequencing is very short, so we just cover a small bit of V segment and CDR3 region. And then it's unclear to me if B cell clone size is at all important, right? So for T cells, it is important. For B cells, I'm not that sure, but I'm not a B cell person. And the question is, so I understand that like pairing and clone size estimation is a TCR repertoire dream, right? But I don't know what exactly is BCR repertoire dream, right? So maybe it's unrelated to this technology. And our next steps, right, of course, is that we added additional automation levels on top, which allow us to run hundreds of samples. And we're working on generating cohorts now. So think like Emerson scale, deep paired repertoire data. We're adapting it for human BCR. We have working mouse TCR system, right, which will be, I hope, published soon. We work on some packages for post-analysis and plotting because this data is kind of is unique in a sense, right? Because there's so many replicates, there are additional things and additional visualizations, additional QC steps you need to do for this. And also preprint contains everything you need to reproduce the method, and all the code is made available on GitHub for pairing. And this is my last slide. If you want to set it up in your lab, where to start? Because a lot of people ask me this question. So first, there's a preprint which contains all the details. Check out the GitHub, and we also deposited process data on Zenodo for all experiments, right? So you could try how it works on your machine and see if it will answer questions you have to your samples, maybe. We will publish a more detailed protocol with the paper. Please be patient. It's just that, developing protocols takes a lot of time. We have our internal protocols, but it's about specific equipment and specific rooms, right? I could share that, but you will need to adapt it for your case. If you have a small pilot project, contact me. We're open to collaboration. And a good place to start is actually with manual 96-well protocol, because there is no special equipment needed. There is no investment in equipment. You just need multi-channel and ability to run PCR and plates. And there is also less investment in primers, because you just need to barcode 96 wells. And finally, full protocol, if you want to run it, it requires non-contact liquid dispensers to make it economically feasible. But there is something called Mantis, something called IDOT, Dragonfly. So there's a bunch of devices out there, and you need something to do 384 to 384 plate copy to transfer from PCR1 to PCR2. But it's not, this equipment is present in many labs. And with this, I want to thank people who worked on this. I want to thank my group, Alison Kirk, Samirat Hikari, Balaji Sundaramaran. And I want to thank Thomas Lab and in particular, Anastasia Minervina and St. Jude Trace study team who collected and processed this COVID sample. And with this, I want to thank all of you and answer any questions. Thank you.
Very nice talk. I would think there are probably many questions, but I will maybe start with one or two questions before others also ask questions. I think maybe I've missed it, but you haven't touched on antigen-specific sequencing. Um, so how, so now, yes, I mean, it's great. You can look at clonal expansion and so on, and then afterwards you can infer with overlapping databases. But can you also do high throughput, large scale tetramer sorted T cells or something? Yeah,
Yes,
probably you have
so
thought about this.
basically, we've thought about this, and we are trying to do this. So the issue with tetramer cells, they're quite rare.
Yeah.
Right? And then there are two actual ways to approach this, right? So we have this protocol when you put one cell per well, right? And actually our problem with 10x and antigen-specific cells is frequently we don't have enough for 10x.
Yes, exactly.
Right? So you run an experiment and you end up like with 300 cells. All of them are nice and precious, but you couldn't run 10x on 300 cells. I mean, it's wasteful, right? And here, this protocol helps in this case because you put one cell per well. And we're working on if you have DNA barcoded tetramers, we're working on a system to amplify the DNA barcode from the same well if you put single cell per well, right? But if you're working with fluorescence tetramers, this is not needed. You could do index sorting, and when I mean, tetramer in this color end up in this well, tetramer in that color end up in that well, right? So this is definitely possible now, and I think this is useful technology here just because antigen-specific cells are very rare, right? The question is, if you can do it in a more high throughput fashion, like, in 10x people barcode their sample with DNA barcoded, stained with DNA barcoded tetramers and then run 10x amplifying both tetramer barcode and alpha beta, I think this might be possible, but I think it will be very noisy. And here the approach would be that you use this tetramer barcode as like a third chain. And you pair alpha to beta to this tetramer barcode. I'm not sure this will work great. I mean, it might in principle, but a lot depends on the efficiencies, right? If you could efficiently amplify barcode, if there is limited noise, because what we learned from 10x, these assays are noisy, right? So each cell is bound by a lot of DNA barcoded tetramers. You need to carefully, so this is the idea here.
Yeah, makes sense. Are there questions from the audience? If not, I just go on.
Oh, I've got a question, if I may.
Yeah.
Hi, Mikhail, that was great. I'm just wondering, in terms of scaling towards bigger cohorts, is there any like cheeky, clever trick you could do by, say, instead of doing one donor per plate, could you mix, so take 4 donors and 4 plates, mix each donor across a couple of plates, and each one's big clones are diluting out its neighbors, kind of thing?
Yeah. So hi, Jamie. Right. So this is great question. Right. So two ways to approach it. Indeed, you could mix PBMCs together. And this is actually what Adaptive did in the original paper when they were proving that, because there were no 10x at the time, right? So this is how we proved that the pairing is sufficiently precise. They mix donors and also sequence them separately, and they show that pairing only happened within a donor, right? You don't have like cross-donor chimeras when alpha is from one donor and beta is from another donor, unless it's like convergent recombination, right? So you can do that. You can run this assay on a mixture of donors, and then you could sequence this donor separately to map the clones back, right, from the mixture. It will work. We don't like to do, I mean, we tried this early on, but what we like to do instead is maybe split a plate into 2 or 4 partitions and do like 192 or 96 wells per individual donor without mixing them together, right? And basically what we're working on is how do we automate the whole process even further. I know, like use robotic hand, which move plate from liquid dispenser to the PCR machine and back, right? How do we make it run more plates per day when we do now? This is the main direction of automation, and this is how we want to move to bigger cohorts, right? Increase throughput in terms of plates, not mixing donors together, because this will require additional experiments to demix them.
Sure, that makes sense. Thank you.
Yeah.
Are there more questions from the audience? If you have a question, please raise your hand via the Zoom thing so I can see. If not, I will ask another question. So, so you mentioned some use cases. Can you explain a bit more? How will you use these data sets? So you showed that you can look at in a paired fashion now. Before we could do it only with bulk sequencing, right? And now we can do it with paired sequencing, the clonal expansion up and down, and finding potential antigen-specific clones. What is the super added benefit to bulk sequencing? Let's say, of course, paired chain information, but what more if we don't have the antigen binding information and not the transcriptome? Yeah.
Yeah, so I think like big picture here is following, right? From in vitro experiments, we know, but if you have alpha beta TCR, right? If you have both chains, you could set up an experiment. You could think like Michael Birnbaum, Chris Garcia, yeast display technology. You could think of like combinatorial peptide pools. So you could think about different experiments, but if you have TCR, you might be able to deorphanize it in vitro without having access to a patient sample, right?
Yeah.
So I think this gives us some, and on the other hand, we know, but if you know just one chain, I don't know such experiments which allow you to do this, right? So it's like having, first half of mystery novel and then trying to guess like who the killer is, right? So if someone else read it, they can tell you, but right, if you're the first person looking at it, right, there's no way to guess, right? And random pair will not work usually if even half of it is right. So I think the hope is that if we accumulate big amounts of this paired bulk, so paired deep data, think like Emerson style cohorts, and we have additional information about exposures on top, then machine learning will start to work. So this is the idea, right? Because in principle, all information about TCR specificity is contained in the paired sequence, right?
Yeah.
Because you could make an experiment in vitro, like figuring it out, right? So probably machine learning model could figure it out as well.
Yeah.
So idea is that just accumulation of this data is useful. And also, I mean, this kind of scenarios when you have a cohort, for example, for a certain autoimmune disease, you sequence everyone deep and paired, and then you look for the shared, TCRs between different people. Usually these TCRs are small, right? So this is why it's so challenging. And when you test them against different antigens you think are involved, or this like longitudinal use cases when you don't know the antigen, but you see something goes up and down, probably it's involved in current infection. But as I showed, it's not that straightforward. I mean, it might be not COVID as you expect, but EBV, right? But again, this gives you opportunity to take next steps.
Yeah, yeah. It would be almost nice to redo the Emerson study again and see if one finds more CMV-associated clones than the 164 that they found, just because it's now
Yeah.
paired or something like that. Yeah.
Yeah, and also in this case, I mean, it's testable now, right? So if
Yeah.
you find something and you say it's associated with
Yeah.
disease, and you go
Yeah.
ahead and test it, right? Emerson sequences, we could not really test them,
No.
right? I mean, they're statistically associated, but it's just features, right?
Yeah.
So we could
Yeah.
not
Yeah.
tell what we recognize, in
Yeah.
fact, easily.
I agree. I agree. I have one more question if there are no more questions. This was a more specific question. I really like that you did in the experiment that you did in the longitudinal one, bulk-seq and then the two single-cell approaches. Did you also, maybe you showed it, correlate the clone counts from the bulk with the clone counts from the single cell, from the TIRTL-seq?
Yeah, I don't show it here.
Yeah.
I did it, right? It correlates. I mean, it correlates nicely, right? So there are
Okay.
some biases, of course, but in general, the reproducibility is good. It's better for beta chain than for alpha chain. The key technology thing, this is V multiplex, right? So you could expect some primer biases. There are no UMIs, right? Because we don't want any cleanups. We want really simple, straightforward technology when you start from cells and don't do any cleanups in process. So this is why there are some simplifications, but it works reasonably well for beta chain if we compare it both to bulk and single cell. So there are no cases when, for example, you lose something, but you have some gigantic clone which is found in bulk but is not captured by TIRTL-seq at all, right? So there are no cases like this. TIRTL-seq primers were specifically depleted for pseudogenes. So if you're interested in pseudogenes, this is not technology for you because we don't amplify this pseudogene non-functional sequences to like simplify and increase efficiency, right? But basically, this is the only difference I see. When I compare bulk to TIRTL-seq, I see good correlation in clone sizes, except non-functional pseudogene clones, which are not amplified by TIRTL-seq primer set.
And one last thing, because I know you need to go also. So you said that for the sequencing, you have very short window on the TCRs, so you don't cover the full VDJ. It would be nice in the future, I think, to have the full VDJ because now we know also through some initial papers that there are polymorphisms in the germline gene. So if we could that associate in the future, once we have much more data of you with cohort, with disease state, I think that would be nice. So yeah, just a comment, not a question. Yeah.
Yeah. But the thing is that sequencing short is cheap and high performant, right? So we do 2 times 150 because this is like what's the most economically. But I mean, it improves with time, right? So just moving the primers left, it's a bit of an issue, but it's not impossible.
Yeah. Great. Thank you very much.
Stop sharing now.
Thank you so much for your talk.